All right, so we're going to start here with the photosystem note. Some of you guys may already have this, so if you do, just fast forward. I think the other class didn't get this. So we've been talking about the components of the process of photosynthesis. What do you have to make it happen? And so obviously you have to have pigments, and a photosystem is that. It is a collection of pigments and some other associated proteins that help collect the sunlight. So in a photosystem, you have really kind of the main pigment, the main chlorophyll molecule, which is what we call the reaction center complex or reaction center chlorophyll molecule. That's the molecule that receives all the energy and sends it out. Around that reaction center complex, though, is a bunch of other pigments that we call a light harvesting complex. Simply a bunch of accessory pigments and proteins that they're associated with that help take all of the rest of the energy that chlorophyll A is not absorbing. They absorb all the other energy and funnel those photons then to the reaction center chlorophyll. When that happens, when all of that energy gets sent to the reaction center chlorophyll, it has to go someplace. And again, this is the difference between chlorophyll and, a synthetic green pigment. Chlorophyll in a leaf, in that thylakoid structure, works with what we call a primary electron acceptor. And so when the electron from chlorophyll A gains that energy and goes to its excited state, right, jumps out of its orbital to its excited state because it's gained all that energy, if there is not something in place to grab that electron and hold it there and, in essence, harvesting the energy, that electron will fall right back down to its ground state, and the energy that it had will be given off as heat or as light in particular fluorescence. And since we don't see plants, like I said before, fluorescing, and you don't touch a leaf and it's hot, obviously they have to have something that they're working with. And the thing that grabs those excited electrons is what we call primary electron acceptors. And so that primary electron acceptor, it's primary because it's the first thing, the first protein to accept that electron, that newly energized electron. And so now that energy from the sun that was passed to chlorophyll A and transferred to the electron of chlorophyll A is now harnessed by that primary electron acceptor, which is your first step in all of the light reactions. And so here we see a photosystem, okay? That purple complex is the photosystem. All within that photosystem are proteins and pigment molecules. So the big purple and lavender things are supposed to be representing proteins. And then you have the pigment molecules that are embedded within those proteins, channeling the sunlight, sending that sunlight then to chlorophyll A, whose electrons are absorbing the energy and, reaching that excited state and moving to the primary electron acceptor. When you talk about photosystems, there are two types. Basically, the only difference is what is the total wavelength of light, or the average wavelength of light that they absorb the best. So photosystem 2 is first in the series of reactions in the light reaction, although it's called photosystem 2. It's only because it was discovered second, but it's photosystem 2, and really the better name for it would be P680. And so there is a chlorophyll A molecule in PS2 that we call P680, and so a lot of times you'll just hear photosystem 2 referred to as P680. Photosystem 1, which happened second in the process, but it was the first one to be discovered, absorbs wavelength of light best at about 700 nanometers. So how do they work? Well, really in the light reaction, there are two ways that the electron can move through the photosystems, and we call it cyclic and linear, or cyclic and non-cyclic. So we are going to start with linear electron flow, or non-cyclic electron flow. Linear makes more sense because you're starting at one place and ending up someplace else. It doesn't cycle back around. So what we're talking about here is the path that the electron takes from photosystem 2 to photosystem 1. It is the primary pathway because it gives us both products, ATP and NADPH, from that one energized electron. So how does it work? First of all, a photon is going to hit the pigment in the photosystem, that energy gets passed from the pigment molecule until it gets passed to chlorophyll A in photosystem 2, or P680, right? P680 is another way to refer to chlorophyll A. That electron, once it gets excited, right, or has gained its energy, that excited electron gets transferred to the primary electron acceptor. And so here we see that happening. The light's hitting your pigment molecules, your light harvesting units, or your accessory pigments. Their energy is being passed eventually to P680, which is our reaction center chlorophyll molecule, and its electrons are gaining energy and going to the primary electron acceptor. So P680 is missing an electron now, and it is a strong oxidizing agent, meaning that it can pull electrons from things. And so what it's pulling the electrons from is water. Okay, water is required for photosynthesis. Our plants need water primarily for photosynthesis because water is the source of the extra electrons, or the fuel that keeps these light reactions going. So water, using sunlight and enzymes that are specific for this process, water gets split and the electrons from hydrogen get transferred to P680, reducing it. Oxygen is a byproduct of this, and it gets released out the stomata. So now we have our electrons from water there being used to replace or replenish the electrons that we lose when sunlight excites P680. So as those electrons get passed to the primary electron acceptor, they will move through the electron transport chain, or we say they fall down the chain, which is just trying to make us remember that they lose energy. So energy gets released by this falling of the electron, and that energy that's given off is used to help pump the hydrogen across the membrane into a space, okay, into that thylakoid space. And so they build up a proton gradient. Couldn't really be out in the stroma because you wouldn't be able to build up much of a gradient because it's just too much space. But if you're pumping hydrogen into that thylakoid space, that's an enclosed environment, and you can build up a high concentration. So the hydrogen get pumped into the thylakoid space to create that gradient. Then once all those hydrogen are there, right, they want to diffuse back across the membrane. Well, the only place they can get back across, because they want to go high to low because there's a lot of them in the thylakoid membrane, but the only place they can go out is through the ATP synthase. Right, so there's my electron transport chain. You don't need to worry about what the letters mean or what the cytochrome complex is. We're not going to get that detailed. But the electron moves from the primary electron acceptor through those electron transport chain proteins, and in the process is going to generate ATP. Now that electron, once it gets through the electron transport chain, right, and it gave off its energy a step at a time, it gets to PS1 depleted of energy. It has very little energy left. However, sunlight is striking photosystem 1 just like it did photosystem 2, and the light harvesting units of photosystem 1 are collecting that sunlight and transferring it to the reaction center chlorophyll of photosystem 1, which is P700. And so P700 takes the energy and gives it to their electrons, and the electrons then from P700 gain that energy and go to their primary acceptor. So there's a primary acceptor for photosystem 2 and a primary acceptor for photosystem 1. So you can see in photosystem 1 that it's the same basic process, right? Light is hitting our light harvesting units. It gets passed to our reaction center P700 chlorophyll. The electron gets excited and gets picked up by the primary acceptor. Okay, now the last thing that happens is that electron that gets excited out of PS1 gets passed off to the primary electron acceptor for photosystem 1. That primary electron acceptor is going to pass its electrons to NADP. NADP is a hydrogen or electron acceptor for photosynthesis. Sounds just like the one we talked about in cell respiration, NAD, right? They're cousins, if you will. They're chemical cousins. They do the same job. They are hydrogen acceptors. So that electron and some hydrogen get picked up by NADP, forming NADPH. And now NADPH is carrying the electrons and the hydrogen so that we can use them in the Calvin cycle. And so there we see our electrons in photosystem 1 that got energized, picked up by the primary acceptor, passed down that short electron transport chain, passed off to NADP, forming NADPH.